monthly live webinar focused on supporting high quality secondary science education and Year 6 classes. Each session brings together experts, and here today we have Edvotek, which I'll introduce properly, Danielle, in just a second. So we're bringing together our key suppliers and science teachers, science technicians and specialist suppliers to share practical insight, curriculum support and real-world classroom applications. At the end, we will send a certificate of attendance and a list of products used as part of this session on a follow-up email. So that'll be nice to cover your curriculum professional development, CPD, and it'll be quite nice to show your departments that you have attended this session. So just a bit of background about me. I've been promoting and supporting our Timstar specialist science brand for almost 20 years. Some of you may know me or have met me or done an event together or a workshop, or I have been in your school delivering sessions or talking to you about your science requirements. Just to remind you, we have 4,500 science products covering chemistry, physics and biology, ranging from our exclusive BMS microscopes, Edvotek biotechnology kits and apparatus, House balances, Lascelles physics kits, glassware, chemicals and so much more. And at the request of our lovely customers, we are bringing back a Timstar catalogue this year, which is great news. We have our fantastic resource hub on the website, written by our content specialist, Amanda, who was a former science technician. There is a wealth of science practicals, funding and buying guides to help you within your roles and to secure extra funding for resources. Timstar, along with our other specialist brands, we have Mod Sport covering PE and sport, we have Gresswell, library, furniture, new build, refurbs and projects, technology supplies, all of our design and technology equipment, were acquired by Commercial Services Group back in February 2025. We can now offer all of our schools a one-stop shop with the addition of our other brands in the CSG portfolio, including KCS Education, who've been established since 1930 and are the leading supplier of educational and early years products to schools and nurseries across the UK. We can provide almost 15,000 products ranging from stationery, curriculum materials and furniture, and we recently acquired Landscapes for Learning, supplying durable and affordable wooden outdoor equipment for nurseries and primary schools. I would ask you to visit our websites for further information on each of our brands and have a look at the fantastic range of products. We have the largest field sales team, a fantastic customer service team, and the most fastest and reliable delivery service that we can offer. We're excited and eager to help and support with your everyday needs and make your life that little bit easier. Enough from me. I am going to hand you over to our lovely Danielle from Edvotek, one of our key suppliers, who will be delivering an exciting and engaging DNA session for you. Any questions, if you could please put in the chat and we will answer throughout the session. Thank you and enjoy. Over to you, Danielle.
All right, so let me see. I will have to be made in, can someone put me as the speaker? Am I set as speaker now?
I am going to put you on as the speaker.
Great. I noticed there were a few people in the chat that mentioned that they can't hear. There is a little next to the mic, there is a little down-facing caret with a drop down. So I would check there is the first place if you're having problems hearing me. I would, again, check there. just to make sure your speakers are on when you turned off the mic. So just for bandwidth issues, and so that uh, no one likes to hear themselves talk, but just so that I can hear myself talk, we'll keep mics off for now. And questions can be placed in the chat as they arise, and I will see them. So thanks so much for having me here. I'm really excited to be able to be presenting this work for our partners at Timstar. My name is Danielle Snowflake. I'm the Senior Director of Education at Edvotek. We are the biotechnology company. We are headquartered in Washington, D.C. It is a snowy morning here in D.C., but I'm really glad to be able to be here chatting with you all today about DNA investigations. So if you're not already familiar with Edvotek, we have been working in the science education space now for over 35 years. It's almost 40 years now, actually. And we were founded in the 1980s by Dr. Jack Trixian. He was a professor of biochemistry at Georgetown University. And what he recognized was there was a lot of really amazing biotechnology, molecular biology, genetic engineering. All these techniques were being performed in the biotech lab. They were not being performed in the education space. And as we know, hands-on inquiry-based learning is a really great way for students to engage with science lessons and to learn about basic biology, higher level biology, and all these amazing things that are happening in the science labs. And if we think about what was happening in the 1980s, I always like to bring up the case of insulin, which is a critical medication used worldwide to treat diabetes. And before the 1980s, the insulin that we used was extracted from animal pancreas. It was unreliable. The dosages could be different. There wasn't a single source of insulin to be used. And so with the advent of DNA sequencing technology, with the advent of molecular biology and genetic engineering, researchers were able to take a human gene, move it into bacteria, and start making a medication that is critical for human health. And so just things like this being translated into the classroom, it really brings a connection to students with what's happening in the real world. And so we're really happy at this point to be working with educators all over the world to bring this hands-on biotechnology to classrooms. And so today in our workshop, we are going to be talking about the ways that forensic scientists might analyze crime scene samples that contain DNA. We're running two separate electrophoresis experiments today. I can hear someone. Can you please make sure your microphones are muted? Thank you. We're going to be running two different experiments simultaneously, both which are simulations of forensic DNA fingerprinting. The kits we're running are both available through Timstar. They are our kits, Edvotek part number S51 and 109. So one of the kits uses DNA. One of the kits is using dyes to simulate DNA in order to look at the mechanisms of DNA fingerprinting. Now, both of these are agarose gel electrophoresis experiments where we're going to be separating these molecules based on their physical properties. For DNA, that's going to be based on its molecular weight or its size. And the dyes are going to be separated by their charges. To be clear, these are simulated samples. There are no hazards. This is not biohazardous materials. These are simulations that are going to be safe to run in your classrooms. And if you're not doing electrophoresis already, and you're worried, we do have all of the safety data sheets available that you can bring to your chemical hygiene officer in order to figure out how best to work them in your space. So by the end of this training, I'm hoping to tell you a lot of exciting things. We'll be talking about forensic science, human genetics, bioethics, as it's going to relate to genetic DNA analysis and our forensic DNA, and the principles and practice of DNA and dye electrophoresis. So to get started, I always like to start with a little introduction to forensic science, because I think many people think of it as a single discipline, when in actuality, forensic science is going to be the application of scientific knowledge from many different fields of science in order to answer questions of interest within the legal system. And again, we're in the United States, things might be a little bit different as we go country to country, but the techniques and the science are going to remain the same. We most often think about DNA forensic science as doing DNA fingerprinting, of crime scene samples, but there's so many more aspects of forensic science that can be applied in your classroom. And all of this data comes together to form the field of forensic science. To answer these legal questions, forensic scientists are going to collect and analyze evidence found at the scene of a crime. They need to keep evidence secured and maintain chain of custody to ensure it hasn't been tampered with by outside parties, or if contamination had come during the collection process, we want to make sure that the evidence that we're collecting is as it was found in the crime scene. Furthermore, forensic scientists need to keep good records of their experiments throughout the entire process because these results are evidence. And that translates really well to your classroom because you are going to want your students to keep a really good lab notebook of all the steps that they're doing and then present it as a lab report. After analyzing the evidence, forensic scientists are going to act as witnesses at trials by providing detailed reports and expert testimony of the analysis that they performed. And so here's a little crime scene we set up in the alley at the back of our building in order to put together a little crime scene to demonstrate where forensic techniques are being used. And again, because forensic science is not just a single discipline, assays from many disciplines are used to analyze the evidence, and they are present here in the slide. And so for example, forensic anthropologists are going to analyze remains that may be found in advanced stages of decomposition. The scientists will look at the shape and the size of the bones to analyze them and collect information. They're going to rely mostly on evidence provided by those remains to determine who died, how they died, and when they died. Next, there is genetics and molecular biology. This is where many of the applications of biotechnology will be found. For these tests, our scientists are going to be analyzing biological material like blood, saliva, urine, and other body tissues to determine where they are and what may have happened. Technologies include the use of antibodies to detect whether a sample was blood, whether it's human blood, and then to test the blood type. In today's workshop, we're going to be discussing the DNA analysis of biological samples and how that information is used. Toxicology is going to be another important field. This is going to identify poisons, drugs, and/or alcohol in tissues and body fluids. This can include drugs that incapacitate people or to show that someone was under the influence of drugs or other substances when a crime was committed. Chemistry will be used in the analysis of trace physical information. Forensic chemists are going to perform analysis of this evidence. It could be drugs, it could be paint, it could be soil, it could be glass or fiber to determine their composition. And then pathologists are going to examine bodies to determine a cause of death post-mortem. In these determinations, they'll analyze wounds, take samples to send to biologists and chemists, and perform histology or analysis of tissues. And again, this evidence is going to help the pathologist to determine a cause and a time of death. So the thread that connects all of these different scientific techniques are the outcomes. Each of these disciplines is going to give us evidence that together can be used to build a case. Today, again, we're focusing on DNA fingerprinting to create a genetic profile from the crime scene evidence. And so I really liked using forensic science experiments in classrooms because I think in addition to being able to teach biotechnology and all these other areas of science, depending on what your discipline is, or if you're an integrated curriculum, we can actually push our student learning beyond just the biotechnology lab. We do have I'm sure you all have many amazing lesson plans available. We have one available on our website where students are going to read witness statements, evaluate alibis before performing the laboratory analysis. And this is a really important exercise to help students develop their critical thinking and literacy skills in addition to their biotech analysis skills. So we want to make sure that we're bringing in evidence from multiple different lines in order to make our final conclusions. And so again, when performing our tests, your students are going to keep careful lab notebooks, documenting chain of custody and the results of their experiments and how they were performed. Plus, if you incorporate some of our other labs, as I mentioned on the last slide, those are going to be additional pieces of data that that can be integrated to work with the results here. And the students need to bring together all of the data from the separate experiments to form a conclusion. And then using the evidence gathered from the case, your students are going to form a hypothesis, and in addition to their lab report, they can write a persuasive essay or make a presentation to the class to build a case for or against, their hypothesis. So they can free the innocent, or they can determine who may have been at the crime scene. And then after performing this investigation and making these conclusions, your students will have experience not only with data collection and critical analysis of results and scientific inquiry, they'll also be able to learn about persuasive writing and clear communication in scientific disciplines. And through my conversations with educators, either at work or at conferences, students are really engaged with these lessons and they have a lot of fun with the whole experience. And so today we're talking about a break-in in a lab. So this is a little scenario that I'll be presenting. You can make this scenario for anything that fits your classroom, fits your neighborhood. We know people who set up crime scenes using their principals and the other staff to kind of create an entire scene. I'm going to just not joke. I'm just going to say that we are investigating crime that happened in a lab. Late one night, an intrepid scientist was working on an important biotechnology experiment in the laboratory. She was very close to creating a groundbreaking vaccine that could save many lives. After working in the lab all day, she decided to go home and eat dinner and get a good night's rest, which is important for all of us. And what you can see is we have a lab bench here. We're going to say that lab bench is after the crime was committed. But if you look at my own lab bench, sometimes you'll see the before and the after aren't very different. However, we will say that the next morning the lab was in shambles. The window to the lab was broken and there was blood on the window from where the glass was broken. The scientist found that many important pages were ripped from her notebook, and that the samples were missing from the freezer. And then upon investigating this crime scene, the police found evidence that they believed to be blood. As forensic scientists, we entered the crime scene and collected the evidence to be analyzed. The sample on the broken glass was identified as blood using presumptive and confirmatory tests. And so now we're going to talk about how we analyze the DNA. In this workshop, we are using agarose gel electrophoresis to analyze our forensics DNA samples. This is a biotechnology technique that uses electricity and a porous gel matrix to separate mixtures of molecules into discrete bands based on their size or other physical characteristics. It's important to know that as the length of a DNA molecule increases, the distance it can travel decreases. This is an inverse relationship. And this slide is going to give us an overview of what electrophoresis looks like step by step. After we created gel, we submerge it in buffer and we load our samples. We attach the safety cover in the leads, then turn on our power supply. After about 20 minutes or so, we're going to check on the results of our experiment. And I'm actually going to start running an experiment right now, so that we will have real authentic results by the end of our workshop together, assuming I set up everything correctly. Science is a human process. I tested everything this morning, and I know that it's working, but mistakes do happen. And so I created the gel, we will submerge it in buffer, attach a safety cover and leads, and we'll run the experiments. So what do we need to perform electrophoresis experiments? So this is where I'm going to switch over to my hands camera because we are actually going to be loading a gel together today. Let's take a look at the reagents that we're using. Always, I want to say, use proper protective equipment. The reagents that we're using today are non-toxic. They are generally regarded as safe, but it is important to always use proper PPE just to, we want to reduce the risks to ourselves and to our students. Again, most of what I'm not using anything today that I would be terribly concerned with touching with my hands, but we always want to get into those good lab safety practices. First, we're going to talk about the samples we're using. Electrophoresis is a very versatile technique. It can be used to separate a variety of molecules, nucleic acids, dyes, DNA, RNA, protein. In today's live stream, we're going to be using it to separate solutions of both dyes and DNA. They come in these quick strip from Edvotek. They come in this quick strip format where they're already pre-aliquoted and ready to go. So you just hand each of your student groups one of these quick strips and the samples are already prepared. It's really nice that they are pre-aliquoted for you. This is going to reduce the amount of lab time you have, the prep time you have before the lab in doing that work. In my chambers, I actually have two chambers here. I have our M12 and I have our edge chamber. I already have the electrophoresis buffer in these chambers. Our electrophoresis buffer is a really important part of electrophoresis experiments. It is a mixture of buffers and salts. And there are two reasons that we use this buffer instead of water. First, electrophoresis uses electricity to separate our molecules, and water is a poor conductor of electricity. So the salts mix with the water, which allows the current to flow through our electrophoresis chamber. Next, we are working with biological samples, and we want to keep them at a pH that does not disrupt their starch, charge, or structure. So when you get an Edvotek kit, you get an Edvotek kit like this in the box. And the nice thing is that it comes with the samples again in that quick strip format. And then it's going to come with the buffer as a concentrate, meaning you just have to dilute it when you get to your classroom. There's no mixing of reagents. It's just add water. Everything comes as a concentrate and it's really easy to use. It's already pre-measured. Agarose is also in this bag. It comes as a powder, pre-measured at 3 grams, which is enough to run 1 classroom worth of gels. Agarose at the molecular level is going to act like a strainer to help us separate our molecules by size. And when we prepare for agarose gel electrophoresis, we need to pour the gel, and I have a little video on how to do that. This is when we prepare the gel, this is kind of the equivalent of making a scientific Jello mold. The agarose you receive in the kit is a powder, much like Jello. We add the powder to our electrophoresis buffer and heat it to boiling that it melts. As the solution cools, it's going to take the shape of its chamber. So we cast the agarose using a mold that fits into our gel chambers. Now, each electrophoresis system is going to have a slightly different casting system, but the principles are going to be the same. So let's go. Oh, did that start again? That did not need to start again. I'm going to take the lid off my electrophoresis chamber. Each electrophoresis system is going to have a slightly different casting system, but the principles are going to be the same. The gel tray has open ends, which allows our current to flow through the gel. But if we tried to pour the agarose solution into this tray, the mixture would leak out, and we wouldn't be able to form our gel. So to prevent this, we have rubber end caps at the end of our gel trays. There's no messy tape or complicated clamps. These rubber end caps that we do include with our gels, they are fantastic. They form a very tight seal, and it allows us to prevent leaks. You just need to be a little bit careful when you pull off the bumper at the end. Just because if you pull it too hard, you can potentially damage the wells. I always like to do this end first, away from the wells, just to kind of get a feel for it. We also have a comb in our gel. I'm going to remove that right now. These are going to create wells where we are going to load our samples. Each tooth on our comb creates a well, and we load 1 sample per well. And you can see our combs come with 6 or 8 wells, which you can choose which side you need, depending on the experiments you're running and how many students you have in your classroom. Once the gel is solidified, again, we are removing the caps and the combs, and we're going to place our gels into our gel chamber. I'm just going to finish doing that on our M12 system. I'm going to be demonstrating 2 different electrophoresis chamber systems today, just so that you can see how 2 of our most popular options work. But again, electrophoresis is going to be the same technique. If you already have it in your lab, we can use the equipment you already have in your lab to do these experiments. The Edvotek reagents will work with any electrophoresis system you have. And if you have any questions, you can contact us, or you can contact our colleagues at Timstar, which will put you in touch with us, and we can help you get the best setup for your lab and, determine which reagents are the best ones to use for the lab. So we are going to load our samples using an adjustable volume micropipette. This is a precision instrument. It is used to make measurements of very small samples. So oftentimes in the laboratory, we are loading very small volumes of liquids. Right now, I'm loading 35 microliters per well. We're loading 35 microliters per well. And at this point, I am putting them into the sample wells. The nice thing about having an adjustable volume pipette, it allows us to make sure our measurements are exactly the same every time. And as long as we're using good pipetting technique, I am pipetting around a camera right now. So normally you want to hold your pipette straight up and down for the most accuracy. However, loading around a camera is sometimes a little bit tricky. But for agarose gel electrophoresis, for these experiments, our samples are incredibly concentrated. Even if you don't get the exact right amount of sample into the well, you're still going to get great results. What you'll notice is that the samples are sinking into the well. All of the Edvotek samples are prepared with loading buffer that contains glycerol, which is going to make the samples more dense than our electrophoresis buffer, allowing them to go into the well. The samples I just loaded were our dye samples. So those are going to be colorful dyes. They're going to separate based on their charge. And we're going to be able to watch that happen, separate in real time. It's great for short class periods because you can run them and see great results in 10 or 15 minutes. The next samples I'm going to load are our DNA samples. And these are going to be, this is one of our DNA fingerprinting kits. And actually both samples that I'm running today are going to give us the same pattern, just one with DNA and one with dye, so that you can see both of the scenarios and determine which would work best with your students and within the constraints of the class or the activities that you're offering. I'm changing the pipette tip between every sample. This is great practice in the lab because it is going to allow us to eliminate cross-contamination, which would happen if it happens in the forensic science lab. It could bias our information. We could be taking one of our suspect samples and accidentally mix it with a crime scene sample, which would be an error that would potentially make it look like someone was at a crime scene when they weren't. In our labs, again, it's really important to have your students keep track of the samples they're loading, which loadings they can load. And if the samples don't match up with what they think they were loading, it's a great time to talk about science as being a human endeavor, how we do make mistakes. And part of the reason we keep notes is to try and figure out where those mistakes are going to come from. Science is messy. And, it is important that students can recognize that even if the results don't turn out exactly as they intended them to, that there is information in that data. All right. Oh, my goodness. Can you all still see me? For some reason, my screen just went blank. I'm going to put the lid on this electrophoresis chamber. I'm going to move it to the side, and I am going to actually start this one while I'm loading our second chamber. So believe it or not, I've got two chambers on my bench today. This is our M12 chamber. The lid is wired with the electrodes, so your students can't actually take off the lid while the experiment is running, so they can't come in contact with any of the current. If the lid is removed, the current is broken. And we do something, we joke, we call the paperclip test, where your students cannot get a paperclip or anything through these vents to actually come in contact with the current. We make this equipment for educators. We want to make sure it's safe and easy to use. I have my power supply. I'm going to set it to 150 volts. I'm going to hit play, and I'm going to have it set for 20 minutes. And we will keep an eye on it during the time our workshop is running. One of the things I always like to say, it's kind of hard to see by video. When you turn on your electrophoresis chamber, always check to make sure you see the bubbles. That's hydrolysis of water happening within the chamber. That lets us know our current is running. The second chamber I'm going to load today, this is our Edvotek Edge chamber. This is an all-in-one chamber. These are very popular right now. They are going to have everything you need to do the electrophoresis experiment in one location. Again, the gel is a little bit longer, but the casting system is the same. Just the gel is a little bit longer. We're going to load this gel really quickly. If there are any questions about electrophoresis right now, I can answer a question if there's anything. Otherwise, I'm just going to continue chatting about the reagents and the resources.
Just to let you know, Danielle, there's no questions in the chat so far. Okay. Does anybody have any questions?
If there's any, I'm just going to load these samples. We have these last 6 samples. Again, I'm kind of loading around the camera. And so you may see a little bit of sample leaking out of the well. Again, our samples are very concentrated, so you are not going to have a problem, even if a little bit of DNA is lost. The other thing I should mention is that while we're running these DNA gels, our agarose gels also have something called CyberSafe included in the gel. This is a DNA stain. It is a fluorescent DNA stain that binds to the DNA in the gel, and it allows us to visualize it instantly when we excite it with blue light. Many times in the lab, people were using ethidium bromide as a fluorescent dye. It allows us to see very small amounts of DNA, but ethidium bromide is identified as a mutagen. And so because it binds to DNA, we kind of expect that. It's an intercalating agent. It gets between the DNA nucleotides. And so a lot of schools have decided to eliminate that from their classrooms for safety reasons. I'm just going to puncture that foil. And so, we use the CyberSafe now. It's excited with blue light, which is also safer for the students and for the DNA. So that's going to be in both of our gels. We do also have a flash blue stain, which I will show you at the end of the workshop. I did my homework yesterday. And ran a gel and got it stained overnight. And that is also an option for running. And so I just turned on our Edvotek Edge. I set it to 150 volts in 20 minutes again. And so we're running both of these gels now, and I will switch between the cameras in order to show you how the DNA and the dyes are progressing over time. But you can see right now, I loaded this and turned it on 3 minutes ago, and we're already starting to get results that we can analyze. And so I'm going to leave this on. I'm going to switch between my electrophoresis systems as we go so that you can see how they are working and how the results are working. But while we're waiting for these experiments to run, I'm going to talk a little bit about the forensic science now. All right, just the little orientation about the equipment again. I'm using the M12 system here. This is for electrophoresis. This is a traditional electrophoresis chamber that it's going to feature two 7 by 7 centimeter gels or one 7 by 14 centimeter gel. We can run up to 2 groups in here at once. I like this system. It's going to look a lot like what you see in a traditional research lab, or you may see this, from other electrophoresis companies. Again, the thing that really sets our equipment apart is that we make it easy and accessible for students to use. We have a rubberized edge group around the side, easy to use buttons to get the lids off. And again, your students can't take the lid off while the gel is running. We also have our electrophoresis edge system. That's going to be an all-in-one system, which combines the electrophoresis chamber, the power supply, and the visualization system. Now, power supplies are generating the current for electrophoresis. Again, here is our power supply. This is going to be our duo source. It runs at 2 voltages. It has a timer, so you don't accidentally run your samples off the end of the gel. This is going to be the perfect power supply for the vast majority of electrophoresis applications. But in case you need to do protein electrophoresis or you want to run more samples at one time, we do have the quadra source as well. Okay, so now back to the science. So as I mentioned earlier, the first thing you do when you find presumed blood samples is to determine whether they're actually blood, and you can do that with both confirmatory and presumptive tests, and there are experiments available for that can work with this experiment. But once you've identified that they're blood, we need to get the DNA out of the samples. DNA can be isolated from any biological sample found at the crime scene. So not just blood, but skin, hair, or saliva. We can't analyze the sample as it's found, though. The DNA is bound in the cell, and furthermore, it's in the nucleus. And as you know, a cell is full of a lot of other components. So here's a very simplified drawing of a cell, but you can see Golgi, lysosomes, mitochondria, ribosomes. There's a lot of other stuff in the cell. So what we want to do is isolate the DNA from all this other intracellular stuff. So first we disrupt the cell walls, and well, there are no cell walls in human cells, but we would disrupt the membranes and in order to release those intracellular components. The resulting lysate consists of our DNA and all of the other molecules found in the cytoplasm. And then using DNA extraction techniques, we'd isolate the DNA away from the rest of the lysate for forensic analysis. So in our case, we're going to say that we were isolating DNA from the red samples that we identified as blood. And if you want to learn more about that, we do have a previous live stream that is available on the Edvotek YouTube page, freely available to watch at any time. And so that could tell you more about forensics blood typing. After the sample is confirmed to be human blood and the DNA is extracted, forensic scientists would turn to DNA fingerprinting to analyze the sample. In humans, our DNA is packaged into 23 pairs of chromosomes. And although this DNA is although most of this DNA is identical between individuals, small sequence differences or polymorphisms are going to occur at specific locations throughout the genome. These are heritable differences, meaning that they are donated from the egg and the sperm, and they are going to segregate in a Mendelian fashion. These polymorphisms include single base pair changes and repetitive DNA elements. And the PCR is used in labs to analyze polymorphisms at several different locations within the human genome. Now, the term DNA fingerprinting is an analogy in itself to describe this biotechnology technique. So if we think about actual fingerprinting, we can take a look at our hands. At the hands are going to differ between people, but generally at the macro level, our hands are pretty much the same. We have 4 fingers and a thumb. They attach to the palm. The palm attaches to our wrist, which attaches to the arm, and so forth and so on. But when we look closer, when we look at fingerprints, then we see a difference. Each fingerprint is going to have a unique arrangement of loops, whorls, and swirls, which can differentiate each of us from one another. In this same way, the combination of polymorphisms can be used to differentiate between individuals. So if we're looking at a position on one of our chromosomes, let's say we're looking at position A, which is going to be on chromosome 2. This is a locus. This is a specific location on the chromosomes. This might be a single nucleotide polymorphism or a SNP. That's going to be a location where the DNA sequence differs by a single letter. I might have an A at that location, and you might have a T, and someone else might have another A. That alone won't tell us much about which of us left the blood behind. In order to create a DNA fingerprint, though, we're going to look at multiple loci within the human genome that are known to have sequence variations. In the United States, the loci analyzed can be searched using CODIS, or the Combined DNA Index System, which is a database of DNA sequences that have been analyzed at these different loci. And the database can be used to compare our crime scene DNA to our crime scene DNA evidence. Each country or commonwealth is going to be different, though. In the UK, DNA fingerprints are created to be run through the United Kingdom National DNA Database, or NDNAD. Overall, though, both databases are going to work in a similar way. We would take our DNA fingerprints that we created in the lab, we would upload them to the system, and then we would use that either to perhaps identify a missing individual or to identify which people were present at a crime scene. And I think it's important to recognize, again, that if we look at just one locus, we could easily have a potential match between 2 individuals, like siblings or cousins. However, when we expand our analysis to look at all of the loci available in the databases, these results become incredibly specific. Since each individual's genome contains a different combination of polymorphisms, we generate a unique DNA fingerprint for that person. If we don't find a match, for an unidentified person or a crime scene sample, we can upload our sequence to the database. If the sample is found at another crime scene, we can link the 2 crimes. This can also use to identify missing people or to rule out suspects later in the investigation. And I'll bring up a little bit about the bioethics of this later, but it's just important to know that at its surface level, we're just looking for specific things within a DNA sequence and trying to match them up. So let's talk about these landmarks that we're using for DNA fingerprinting. We already talked about single nucleotide polymorphisms, or SNPs, in the last slide. That's where the DNA base pairs at a specific location show variability. Now, some of these DNA sequences are defined as restriction fragment length polymorphisms, or RFLPs. And these are the original polymorphisms that were identified and used for analysis in the UK, which is where the technique actually was developed. And so what does this mean? We'll step back a second. So a restriction enzyme is a highly specific enzyme that cuts DNA into fragments based on its DNA sequence. I like to think of them as molecular scissors, but unlike our paper scissors, which can cut any paper, these molecular scissors can only cut at specific locations, and the sequence has to be exact to its match. For example, the restriction enzyme EcoR1 cuts DNA with the sequence GAATTC. Let's say that second A in the sequence is where a SNP can occur. It can be an A or a T. The enzyme will only cut when there is an A in that location. And so if we look at the schematic I have up here on our slide, we have a segment of DNA that is between two black arrows. There are two potential rift lips here, one indicated by the yellow arrow in DNA sample A and one by the red arrow in DNA sample B. If we were just to isolate this DNA and run it on a gel, the DNA samples would look the same. It's the same size of a piece of DNA. However, when we cut with EcoR1, we see different results. For sample A, the cut site is closer to the middle, so our DNA fragments in lane A, you can see it on this gel, they're going to be closer in size, and we see the bands are closer together. In sample B, the cut site is closer to the end, so the two fragments are more different in size. The differences in the pattern would allow us to distinguish between people and between DNA samples. Now, the more commonly used kind of rift lip today is going to be our variable number of tandem repeats, or VNTRs. A VNTR is a short repetitive DNA sequence. The sequence itself is generally between 15 and 35 nucleotides long, though they can be as short as 2 base pairs and as long as several thousand base pairs. And they can occur between 5 and 100 times at a specific location. VNTRs are a type of rift lip because they actually change the restriction pattern by changing the distance between two identified enzyme sites. So before we were changing the sequence of the EcoR1 recognition site to change the pattern, now we're actually changing the distance between the two EcoR1 sites. So if we look at the schematic, in this schematic, we're actually looking at 2 chromosomes present in a person's DNA sample. Again, these are heritable. So one of these chromosomes would be present in the egg and the other in the sperm. And what we can see is that there is a VNTR site between these two EcoR1 sites. There are many more copies of the VNTR present on copy B than on chromosome A. So if we ran this sample on a gel, we would see 2 distinct samples present in just 2 distinct bands present in just one sample. And that's what we're looking at on the gel that we see on this slide. And so what you can see is that we have 5 different DNA samples from 5 different individuals. Each of those are going to give us each of the lanes on this gel are different from different people. And you can see the variety of sizes between the 2 chromosomes, but also between individuals. And so this is actually our experiment 3334, which is a human PCR experiment where the students are analyzing their own DNA. And your students can actually do this analysis in the classroom if you have access to a PCR machine. And so you can imagine how powerful this technique is for DNA analysis, because now we're not just dealing with cut versus non-cut DNA. We have DNA fragments that can vary in size dramatically based on the number of VNTRs present within a single location in DNA. Furthermore, since each person has 2 chromosomes and each chromosome has a different number of VNTRs present at a single location, the amount of DNA that we can get from a single place is incredible. And if we multiply that over all of the different locations used for analysis in our DNA databases, you can imagine why DNA fingerprinting is so specific and can identify individual people. In today's forensic science laboratory, rather than cutting out this DNA using restriction enzymes, we're going to amplify DNA using a technique called the polymerase chain reaction, or PCR. Just right now, though, I want to take a little quick break before we're talking about PCR. I want to make sure to point out that you can start to see really great separation in our DNA samples. These have been running for 15 minutes so far. And I'm going to turn on the sample in our edge chamber. And you can see we're getting great separation there as well. You can see the bands, they're glowing bright green on a lighter green background. I'm going to turn one of my lights off though, so you can see that second band a little more clearly. And so you can see this is running in about 10 minutes. We're already getting separation of our DNA that can be used for our forensic analysis. This bottom gel is also a DNA gel. And I'm going to show you a really great piece of equipment that we have to visualize that lower gel in our M12s in a little bit. So we're going to just keep these gels running while we're going. And then we will analyze the patterns together. And so back to PCR. Again, in our forensic science labs, you're going to be doing DNA amplification to analyze these samples. PCR is a really incredibly powerful technique that takes a small amount of DNA and expands it to be used for analysis. It's not a perfect analogy, but you can think of it like a DNA photocopier. And so PCR actually follows the same steps that our cells use to replicate DNA. And so when I personally teach PCR, I always like to teach it in the context of DNA replication. So first we have to separate our two DNA strands. In the cell, we have the enzyme helicase that unwinds the DNA. In PCR, we heat the samples to 94 degrees centigrade, which is going to separate the double-stranded DNA. The high heat breaks the hydrogen bonds between the DNA base pairs, and that's known as denaturation. Next, if we were in the cell, the enzyme primase would come along and build short RNA primers along the DNA strands. This allows the enzyme that builds DNA, our DNA polymerase, to start catalyzing the bonds between DNA nucleotides. DNA polymerase can't build DNA on its own, though. It needs these primers to get started. In PCR, we're going to design these primers to bind and target specific DNA sequences. So those are going to be our different loci within our human genomic DNA. We adjust the temperature of our samples, which allows the primers to anneal to complementary base pairs based on the interactions between the DNA base pairs and base pairing rules. Once the primers are bound, DNA polymerase can start building DNA using each strand of DNA as a template. In replication, this would be copying the entirety of a chromosome so that we each end up with a perfect copy of our 46 chromosomes, the 23 pairs. In PCR, we are going to raise the sample temperature to 72 degrees, which allows the polymerase to attach to our primers and fill in the molecules, which allows the polymerase to create a section of our original DNA molecule. Now in replication, we are done until the cell divides again. For PCR, however, we're going to repeat these steps 25 to 35 times to create a ton of DNA. We use a special machine called the thermal cycler to automate changes in temperature. In the early days of PCR, this was done by having a grad student move tubes between water baths. So you can see how far we've come to where students can actually use PCR in their classrooms and do the experiment. I think it's a really amazing way to explain how quickly the technology has expanded. This is a technology that was, developed in the late 80s. It won the Nobel Prize in the 90s, and today we're doing it in the classrooms of students. And it's also, you can also start talking a little bit about mathematics with your students when it comes to PCR, because each cycle is going to be doubling the amount of copies of DNA in the tube. And so it's an exponential growth process. And so if you want to bring math into your classroom, you can have your students calculate the number of DNA present in the sample after PCR. So if we start with 5 copies and cycle for 25 cycles, how much DNA is present at the end of our experiment? All right, so let's actually look at some results. This is not the experiment that we're doing today. This is another one, another DNA fingerprinting experiment that's very common that Edvotek offers. So we can say that, this DNA was found at a crime scene and analyzed using DNA fingerprinting. At this location, in this experiment, we're only looking at one locus in the genome, one location, which is from 3 different suspects, and we're comparing it to crime scene DNA. So if we're just looking at the results from this experiment, we would say that the DNA in lane 2 from suspect 2, which is in lane 4, is going to match our crime scene DNA, which is in lane 2. However, since we're only looking at one spot within the genome, we wouldn't be able to make any conclusions from this evidence about suspect 2. But since they don't match at this location, we can rule out suspects 1 and 3. We'd have to look at additional loci before making our final conclusion. DNA can also be used to determine paternity as well. And so also we can use it to look at a new application of the technology, which is going to be forensic genealogy. So remember, our person's DNA is going to be inherited from both parents, 50% from the egg and 50% from the sperm. And using Mendelian genetics, we can calculate the probability that each offspring will have a specific genetic combination. So in this combination, in this pairing that we have on the screen, what we can see is that we have 2 parents that are heterozygous at this one location. So each has an orange copy, each has a white copy of the gene. And that means that each child has a 50% chance of receiving a specific chromosome from their parent. We have a 25% chance for each child being homozygous for either allele, and a 50% chance for them to be homozygous. And so using these genetic principles, a person's parentage can be determined from a child's DNA profile. For example, here we have a simulated paternity test. We have the mom in lane 2, the child in lane 3, and 2 potential fathers in lane 4 and 5. By comparing the DNA profile of the mother and the child, it is possible to identify which DNA fragments in the child are absent from the mother and must have been contributed by the father. And so looking at these bands and looking at the 2 potential dads, we can rule out dad number 2, which is going to be in lane number 5. Excuse me. Dad number 1 contributes the bands in the child's DNA sample that aren't represented. Now, one awesome thing about DNA sequencing technology is that it's getting faster and cheaper. Instead of just sequencing DNA for paternity or forensics or for medical needs to determine rare genetic disorders, people can sequence their own DNA to learn more about their ancestry, their heritage, and their lineage. Certain genetic markers can be learned to link to certain specific ethnic groups. Plus, given that these markers are inherited, we can use them to identify family members. And so in 2010, a group of amateur genealogists created a database for people to upload their DNA sequences. These were to be used for people to identify family members, either near or distant, using markers in their DNA. To this date, this DNA database called GDmatch has over 1.2 million DNA profiles. Now, while most people were using this for genealogy, detectives in California decided to use the database to help generate leads for a cold case. There was a criminal in California who was responsible for multiple burglaries, sexual assaults, and murders from the mid-70s to the mid-1980s. And this person was the Golden State Killer. There's a great book on this case. And this was before DNA technology. So the perpetrator didn't try to prevent leaving DNA evidence behind. For years, the detectives could link the cases together based on the DNA, but there were no matches because this person had stopped, they're performing these acts of violence. But there was no matches in codons. So detectives were at an impasse. They took a DNA sample, had it sequenced, and then uploaded the sequence to GEDmatch. It turns out that the family members had uploaded their DNA sequences to genetic genealogy sites. This allowed the detectives to identify potential suspects, get warrants for their DNA, and then to make an arrest, which is risen in a guilty plea. And so this picture shows the genetic detective, Cece Moore. She is perhaps one of the most famous genetic genealogists and has a whole TV show that has been based on tracking down people, including birth parents, long-lost family members, using these DNA databases. But one of the reasons why I think this is so important for you to cover in class is that it provides an area to discuss bioethics, as it may be related in your students' daily life. They may see it on the news. Because this allows us to discuss important topics. For example, is it ethical to use this information to identify potential suspects? Do your family members have the right to privacy if you upload your DNA information? Forensic scientists have the right to access this data? There are no right answers necessarily to these questions. Everything has nuance, but it is a place for your students to read the literature, to watch the shows, and to form an opinion. And this again can be used to write a persuasive essay, again, bringing in those 4 literacy skills. So we have about 5 minutes left, which is perfect because we're going to wrap back up to talk about what's happening in our gel while they are running. While we were here, we loaded the gels. I prepared them before the workshop. We're loading them and we're running them to separate the DNA samples. Before the workshop, I cast the agarose gels. That was our science jello. We placed the gel into our chamber and then started going. So let's talk about what is actually happening inside of our gels. So we load our samples into the wells. We apply the samples and we apply current. So in this experiment, agarose gel DNA electrophoresis is being used to separate our DNA fragments by size or our dyes by charge. Due to the chemical structure of DNA and these molecules, our molecules are going to be negatively charged dyes. Our DNA has a sugar phosphate bond that holds the nucleotides together. Both of these molecules have a strong negative charge. So in our gel, we locate the wells of the gel near the negative electrode. So you saw when I loaded them, I loaded the top of the gels. Here's our positive electrode. Our negative electrode is at the bottom. We load the samples and we turn on the current. The current is going to push the samples through the gel to the positive side of the gel, from the negative side of the gel to the positive side of the gel. Now let's talk a little bit about the gel. At first glance, agarose gel appears to be solid at room temperature. If you were to pick up this gel, it would feel like a thick jello. It's still soft, but it's less jiggly than dessert. However, at the molecular level, the gel is full of pores, which are little microscopic tunnels that are going to go through the gel. These tunnels are going to affect how different sized molecules separate into bands. As the current is pushing and pulling the fragments through the gel, the molecules have to find their way through these pores. They may have to twist or turn to get through these channels. Since it's easier for the small molecules to move through the gel than larger molecules, the DNA fragments are going to separate into bands by size, with larger molecules near the wells and smaller molecules further down the gel. And so here's a schematic, and we will examine that on our actual gel. Because the molecules are going to travel at different speeds, they form bands. And so if we're looking at an analogy for electrophoresis, we can think about DNA molecules like fish swimming down the river. We have some fish that are large and some that are small, and they're moving downstream with the current. However, the river, as you can see here, is very rocky. You may have rocks or other obstructions like a net in the river. Smaller fish, minnows, will be able to navigate through these tight twists and turns or fit through holes more easily than larger fish. This means the smaller fish will travel down the stream more quickly with the current than the larger fish. We can think of the fish as DNA molecules, the current of the stream as an electrical current, and the obstacles as an agarose matrix. So if we look at the results of this DNA electrophoresis experiment, we find smaller bands at the bottom and larger bands at the top. We always run a molecular weight marker with DNA bands of known size so that we can determine the sizes of unknown fragments in our experimental samples. But we have one more problem. DNA is clear and colorless. We cannot visualize these bands with the naked eye. So we need to figure out how to visualize it using another DNA stain. There are many DNA stains available for use in the classroom, but I'm going to talk about my two favorite, which are CyberSafe and Flash Blue. I'm going to turn off this gel right now. And we'll start looking at these samples. So I have CyberSafe in these gels. Here's our dye gel, and we'll look at it again a little bit more closely later. Here, our second gel, we're going to have CyberSafe in it. I will show you on the next slide what this piece of equipment that I'm sliding under is right now, but this is our TrueBlue Junior. This is a portable DNA transilluminator. It's going to use the blue light to excite the molecules. And so as you can see, we're not seeing anything but our blue tracking dye that lets us know how far the gel has run. I'm going to put this amber filter on and turn on my TrueBlue Junior. And what you can see is that we can see those bright green DNA bands. So that's an example using CyberSafe. However, not everybody has the blue light transilluminator in their classroom. Not a big deal. We also have a stain that allows you to visualize the gels using visible light. It's Flash Blue. I did my homework last night and stained this gel. This is a Flash Blue gel. It's going to be the same banding pattern. Let me take it out of my little fast food container so that it's easier for you to see. Let's move this here. My camera is not moving the way I was hoping it would move. So we're just going to put this right here. I'll put on more light for it. And so this would look better on this looks better on a white light transilluminator. But you can see, I have dark blue bands. I'm just going to put it right here on my table. Dark blue bands on a white gel. You can start to see them there. My camera will focus. We have these bands, the dark blue bands on a light blue gel, and they're going to have the same banding pattern as the bands that I ran here. Let me turn this camera back around. So those are our two DNA stains. I like to use the overnight process for Flash Blue. I think it gives better results. The results are clearer, the bands are darker, and the background is lighter. But here is our CyberSafe stained gel with the same results. CyberSafe, you add it to the gel, so you're actually staining the DNA molecules while the experiment is running. We add the concentrated stain to the gel, run the samples through the gel, no post-staining or destaining necessary. We have two different transilluminators. If you use a traditional electrophoresis chamber, We have the True Blue Transilluminator, which is going to be a system that you put all of your classroom set of gels on. We have the True Blue Junior, which fits under the electrophoresis chamber to allow for real-time visualization. The nice thing is that it also comes with a separate orange hood now, so that we can move the gel out of the chamber, and we can visualize it separately on a mini transilluminator. And again, those are going to be our green bands. Here is our edge unit, which has been running as well. And you can see, again, the same dark green bands on a light green background. Those are our DNA patterns. Now here are our dyes. Again, they don't require any additional visualization equipment in order to be able to see the results. And so let's analyze these results. Again, both gels are going to have the similar banding pattern. We did that on purpose so that you can always use the similar lesson for both classes. And what we see here on the gel and in there is that we're going to be able to analyze these results. What you'll see is that the samples in each lane created different patterns. So in this example, if we're looking at the DNA from our different loci, or we're looking at it as the dyes or the DNA, what we can see is that each lane has a different pattern. In this example, the DNA from two different loci or places in the genome are being analyzed. We'll call them A and B. On this electrophoresis gel, we can see the results. The lanes look different in that all three, the crime scene sample and the suspect samples, all have the same pattern in loci A, but we can see that the crime scene sample matches suspect 2 at loci B, whereas it does not match suspect 1. And so this is really important because this allows us to rule out suspect 1 because their banding pattern doesn't match at that second position. From this experiment, it looks like suspect 2 may have been at the crime scene before and contributed a sample. If we were just looking at loci A, we would not be able to make any conclusions from this evidence. And I think this is a really important way to emphasize how you need multiple pieces of DNA evidence, multiple different loci in order to be able to make a determination. But not only that, that this DNA profile alone does not provide evidence that someone committed a crime. They could have just been there at a different time. And so this is where some of those critical literacy skills, with suspect alibis and other documentation comes in, other pieces of evidence comes in and is important. So we are over now. Thank you so much for sticking with me till the end. We've covered a lot of information over the course of this experiment. I'm just going to recap, and if there's any questions, feel free to put them in the chat now. But just in brief, forensic science is an application of scientific methodology to answer legal questions. The tools for forensic analysis are drawn from many different scientific disciplines, including biotechnology. We discussed how DNA can be analyzed and used to identify family members or suspects or exonerate suspects in a crime. DNA fingerprinting identifies heritable differences in an individual's DNA. And we can use PCR to amplify regions of DNA that vary between individuals, and then use electrophoresis to rapidly separate the molecules to analyze them. And while fairly simple, we just did this experiment together in my basement. It's very powerful when it comes to rapidly separating different molecules from one another. But I do think it's important to remember that techniques like forensic genealogy, while exciting, bring up ethical questions that can be addressed in your class. Data from a crime scene can suggest that a suspect was at a crime scene, but the data alone cannot convict a person of a crime. Many lines of evidence, including witness statements and alibis, must come together to build a case against a suspect. And these results are used as evidence. And so this is Edvotek. The best way for us, this is a lot of our resources that we have available. Again, we also work closely with Timstar to distribute our product in the UK. And if you have any questions, you can get in touch with your representatives there, and they can get in touch with us for any questions. We will be able to share the slides. You will be, okay, great. Yes, that is mentioned in the chat. You will receive a recording of today's session along with the slides. You know, Edvotek is looking forward to being back later in the year with a workshop that dives in more closely to PCR as well, to talk more about the, you know, advanced DNA technologies. And I hope that if you're not already doing electrophoresis in your classroom, this workshop convinced you that is something you can do. I right now am doing this in my basement. It is snow day. I'm very grateful that you didn't have to hear my kids running around upstairs. So thank you to my husband for keeping them quiet. So yes, so if I can do it in my basement, you can do it in your classroom. Please let us know how we can help you make the electrophoresis a reality for your students. Thank you.
Just want to say thank you very much, Danielle, for a wonderful and informative DNA fingerprinting session. That was absolutely fantastic. So well done for that, and well done for being our very first supplier to do one of our webinars.
I'm always happy to be your canary in the coal mine.
Yeah, thank you very much for that. That was so good. That was absolutely brilliant. Just want to say we'll be running more Timstar webinars. As Danielle mentioned, she will be back later this year. So we'll have one every month going into 2026. Our sessions will be covering physics and chemistry. This was obviously very sort of biology focused today. And just a reminder for everyone that we are doing Timstar Tuesday discount days, offering 10% off online orders. So if you need any of these lovely Edvotek products or anything else within the Timstar range, just get online and get those orders through on the Timstar Tuesday discount days. I hope you enjoyed the first webinar session, and thank you for joining us and taking the time out of your busy day. And if there's any other questions, I'm just having a quick look through the chats. Everybody's saying thank you, Danielle. So thank you very much. And yes, there will be a recording of this session. So for anybody that missed it or wants to view it in their own time, it'll be on the Timstar website. And I think I've covered everything. So thank you very much again. Thank you, Danielle.
If there's anything else your educators want to learn about that in terms of biotechnology that we can help with, please be sure to let Lucienne know, and we will be happy to program that content as well.
Excellent. Thanks very much. Thanks again. Take care,
Bye
everyone.
now.
Bye.